Hey everyone, especially those of you fresh out of grad school or just starting your PhDs in biotech. Jumping into this field, it's like drinking from a fire hose sometimes, right?
It is definitely a lot to keep up with.
Yeah, so much is happening so fast. You need to be on top of the latest, but also like really understand it, not just skim the surface.
Absolutely. I mean, who has the time to read every single paper that comes out, right?
Exactly, that's where we come in. We're giving you the crucial insights from cutting-edge research without the information overload. Think of this as your deep dive into the stuff that really matters for you.
We cut through the noise and get right to the core of what's important and hopefully make it a little bit fun along the way.
Exactly. And for today's deep dive, we're looking at a game changer, I think, for anyone working with photosynthetic microorganisms, your algae, your cyanobacteria, those sorts of things.
This is a new automated high throughput lighting system, and it's a big deal, especially for those of you in biotech who are focused on biomanufacturing or sustainable production.
We've got a great source for you today, a really in-depth article from a Nature Portfolio journal. It goes through how they developed this system and all the testing they did to make sure it really works.
It's a fantastic resource, and we're going to break it all down for you today.
So by the end of this, you'll know exactly what this technology does, why it's so important for biotech, and how it could actually change the way you do your research.
Especially if you're interested in harnessing the power of these little green factories for things like producing biofuels, bioplastics, or all sorts of other valuable compounds.
Right, so let's just jump right in. Photosynthetic microorganism, I feel like we hear so much about them, the potential, right?
Oh, absolutely. I mean, they're amazing, right? They can take sunlight and CO2 and turn it into all sorts of useful stuff. They're like nature's little biofactories.
But I feel like there's also been this gap, right, between the potential and what we've actually been able to do with them in the lab and then at larger scale.
Yeah, there's definitely been some roadblocks along the way. They haven't always been the easiest organisms to work with.
So what are some of the main challenges, let's say, compared to working with something like E. coli or yeast?
So there are a couple of big ones. For one thing, the genetic tools for engineering these organisms haven't been as developed as for the more traditional lab workhorses like E. coli or yeast. It's getting better, but it's still lagging behind a bit. And then the second big thing, which is what this new lighting system is all about, is the lack of really good high throughput cultivation platforms, especially at lab scale.
Okay, so with E. coli, you could just, give it some sugar in a flask, shake it around, and it's happy. But with these guys, it's all about the light, right?
Exactly. Light and also gas exchange. You need to be able to precisely control and deliver light to lots of cultures at the same time, and that's not so easy to do when you're trying to scale things up.
Makes sense. So the article actually mentioned a few strategies that researchers have been using to try and get around these limitations. What are some of those?
Yeah, so one approach is what's called pooled selection. Basically, you take a huge population of different strains, grow them all together under specific conditions, and see which ones survive and outcompete the others.
Survival of the fittest, right?
Exactly. It's really useful for screening massive libraries of genetic variants, like if you've done a bunch of random mutations or something. But the downside is it only works if the trait you're selecting for actually gives the organism a fitness advantage. And you can't really control the growth conditions for each individual strain very precisely.
Right, like you throw them all in the deep end and see who can swim. Interesting, but not exactly subtle. What else?
Another approach uses microfluidics and flow cytometry. So this lets you encapsulate single cells in tiny droplets or sort them based on certain characteristics, like how much of a particular fluorescent protein they're producing. You can get really high numbers of individual cells this way.
Sounds high-tech, but are there downsides to this as well?
Well, one issue is you tend to get more false positives. Just because a cell looks promising in a droplet doesn't guarantee it'll perform the same way when you try to growing up in a regular culture. So there's a lot of downstream work to actually isolate and validate those hits. It's a bit like panning for gold. You might find a few shiny flakes, but you need to dig a lot deeper to see if you've hit a real vein.
And then there's that third approach, right? The microplate or microphotobiorreactor systems. I feel like that's probably the most common way people are doing things right now.
Yeah, these give you intermediate throughput, so you can test hundreds or even thousands of cultures in parallel. And you can test different media compositions or growth conditions on clonal populations, which is a big plus.
But they're not perfect, are they? Otherwise, we wouldn't be talking about this new lighting system.
Right. One of the biggest limitations has been the lighting systems for these microplate setups. A lot of them have been either very specialized and low throughput, or you have to manually move the plates around under the lights, which is not very efficient and makes automation difficult.
So it sounds like there's a real need for something that can bridge the gap between high throughput and the kind of automated workflows that everyone in biotech is moving towards.
Exactly, and that is where this new system really shines. It's basically a consistent lighting system that's specifically designed to be integrated with standard laboratory automation infrastructure. Think liquid handling robots, plate readers, all that good stuff.
So it's not just a standalone piece of equipment, it's meant to work with all the other automated systems you might already have in your lab, which is cool.
Yeah, and the big advantage of that is you can suddenly scale up your experiments massively. They tested it with a Cytomat 5C450 incubator, which is a pretty common piece of equipment, and they were able to run parallel screens on hundreds or potentially even thousands of cultures at the same time.
Wow, okay, that is a lot of algae. And is it limited to just certain types of organisms or?
Nope, it's pretty flexible. They've shown it works with both prokaryotes like Synechococcus elongatus and eukaryotes like Chlamydomonas reinhardti. So it covers a pretty broad range of photosynthetic microorganisms that people are working with in biotech.
Okay, so it sounds impressive, but I'm guessing it's not quite as simple as just slapping some LEDs onto an incubator and calling it a day. There must be some pretty specific design and engineering challenges that had to be overcome to make this work.
Oh, yeah, for sure. To make a system that can really reliably support photosynthetic growth at this scale, you have to think about a lot of different things.
So what were some of the key things the researchers focused on here?
Well, one of the absolute fundamentals is making sure the light distribution is even and consistent across all the wells of a microplate. You don't want some cells getting blasted with light and others barely getting any.
Right, that would totally mess up your results, right? You'd be comparing apples and oranges and think you found something interesting, when really it's just a lighting artifact.
Exactly. So they set a pretty ambitious target for themselves to achieve less than 5% variability in growth under conditions where light isn't the limiting factor.
5%? Wow, okay, that's pretty tight control. But why that specific number? Is there like a biological reason for that?
Yeah, there's actually some cool science behind it. To figure out how important consistent light really is, they used what's called a protein economy model of this cyanobacteria called Synechocystis sep, PCC 6803. Have you ever heard of those?
Protein economy models. Rings a bell, but maybe you could remind us what those are all about, especially in the context of making sure we get reliable results for biotech applications.
Sure, think of it this way. Imagine a cell as a factory, right? It has limited resources like energy and building blocks, and it has to allocate those resources to different tasks like growing, repairing itself, making stuff, you name it. These models help us understand how cells prioritize those tasks depending on the resources they have available, including light.
Okay, that makes sense. Priorities change depending on what's available. But how does that relate to this 5% variability target?
Well, what they found with the model is that when light is scarce, the concentrations of different proteins and metabolites inside the cell fluctuate a lot more. But once you get above a certain light level, kind of where growth starts to level off, then minimizing variability in the light becomes really important for keeping the cells stable and predictable.
So by making sure the light is super consistent, you're basically ensuring that all the cells in your experiment are behaving in a more uniform way.
Exactly, which is super important for getting reliable and reproducible results, especially if you're trying to optimize the production of some valuable compound or engineer a strain to do something specific.
Makes total sense. So consistent light is key. Check. What else is important when designing a system like this?
Another big one is adjustability. You need to be able to tweak the light intensity to match the needs of different organisms and experimental conditions. So their system can handle up to 50, that's right, 50, 385 well plates at once.
50 plates. That's amazing. How many individual cultures is that? Thousands.
Yeah, I think it works out to something like 19,000 individual cultures you could potentially be growing at the same time, and you can adjust the light intensity for each plate across a pretty wide range from 50 to 500 ohmolars Sarper.
You got it. It's basically a measure of how much photosynthetically active light is hitting a given area per second. So this system gives you a lot of flexibility to really dial in the perfect light conditions for whatever you're growing.
Okay, so you've got consistent light, you've got adjustability, but then there's the whole question of making this thing actually work in a real lab, right? It has to be compatible with all the other equipment and robots people are already using.
Right. You don't want to have to redesign your whole lab just to accommodate a new piece of equipment. So they made sure the system adheres to the standard form factors defined by SLAS, the Society for Laboratory Automation and Screening.
SLAS. Okay, so that means it should basically just plug and play with most of the common lab automation systems out there. Pretty much. And they also paid a lot of attention to practical considerations like modularity, using readily available parts, making it easy to clean and maintain. Things like that can make a huge difference in day-to-day use.
Yeah, especially in a biotech setting where sterility and reproducibility are so important. Okay, so high-level design goals, check. Let's get down to the nuts and bolts. What are the core components that make this lighting system tick?
So at the heart of it are these metal plate-based LED arrays. Each one has 384 individual LEDs that are precisely aligned with the wells of a 384 well plate. They used simple positive and negative connections for each array, which makes it easy to scale the system up if you need even more capacity.
So each little well gets its own dedicated LED. That's pretty cool. But LEDs generate heat, right? Did they have to do anything special to make sure the temperatures stayed consistent across the whole plate?
Yeah, heat management is really important, especially with sensitive organisms. They actually did a whole bunch of testing to figure out the best way to deal with the heat generated by the LEDs.
Because, yeah, too much heat can stress the cells out, mess up your growth rates, all sorts of problems. What did they do to minimize temperature variability?
They actually compared two different materials for mounting the LED arrays. One was copper, which is a pretty common material for heat sinks, and the other was this superconducting aluminum alloy. And they carefully monitored both the light intensity and the temperature across the arrays over a pretty long time to see how stable they were.
And what did they find? Which one performed better?
The aluminum alloy blew the copper out of the water. It had way less variability in both temperature and light intensity. And this was true even when they cranked up the power to the LEDs.
Wow, okay, so this fancy aluminum alloy is really a key part of making this system work so well. What was going on with the copper? Any ideas?
That's the interesting part. With the copper-mounted LEDs, they found that the temperature and light intensity were actually correlated, meaning that as the temperature went up, so did the light intensity. Which is kind of weird because usually you'd expect hotter LEDs to be a bit less efficient, so they think there might be some fluctuations in the electrical current with the copper arrays that could be causing both the light output and the heat generation to go up and down together.
Interesting. So sounds like they made the right call going with the aluminum alloy. Consistent light and consistent temperature. That's the name of the game. Okay, so they've got the hardware figured out. What about actually making sure it works with real, live photosynthetic organisms?
Yeah, they didn't just stop at the hardware testing. They did a ton of experiments with Synechococcus elongatus to really optimize the whole system for supporting photosynthetic growth. And they looked at a bunch of different factors, like the light spectrum, the types of seals they were using on the microplates, how close the LEDs were to the cells, even the type of microplates themselves.
So like a full-on optimization process to make sure everything is working together perfectly. Let's start with the light spectrum. I know different photosynthetic pigments absorb different wavelengths of light, right?
You got it. So they tested LEDs with three slightly different color temperatures, 4044K, 5091K, and 6083K. Each one has a slightly different balance of wavelengths in the visible spectrum.
And which one turned out to be the winner for Synechococcus?
The 5091K LEDs gave the best growth, they think, because that particular spectrum provides the most even spread of light across the wavelengths that Synechococcus uses most efficiently for photosynthesis. Of course, this might be different for other organisms, so it's something to keep in mind if you're working with a different species.
Good point. Okay, what about those seals on the microplates? I'm guessing that's not just about keeping things from spilling.
Oh, it's way more than that. Those seals can have a big impact on things like evaporation and gas exchange, which are super important for how well the cells grow.
Makes sense. So which seals did they test out?
They tried a couple of breathable seals, one called AeroSeal and the other called Breathe Easy, and then they compared them to just regular transparent lids. The AeroSeal, which is made of this opaque woven rayon material, actually gave the best results overall.
Opaque. That's interesting because the light's coming from below, right?
Yeah, it does seem a little counterintuitive, but they think the opacity might actually help to minimize light scattering and create a more stable environment inside the wells. Plus, AeroSeal is really breathable, which helps with gas exchange.
But didn't they run into a little snag with AeroSeal?
Yeah, unfortunately, it's not ideal for automated systems. Robots have a hard time gripping those woven rayon seals, so they ended up going with another breathable option, PermaSeal, which is made of woven paper. It worked almost as well as AeroSeal, and it plays nicely with their robots.
So a good compromise between optimal growth conditions and automation compatibility. Did they track how much liquid was evaporating from the wells with these different seals?
Oh yeah, for sure. They found that the PermaSeal had the highest evaporation rate, about 57 microliters over a 96-hour period, which is something to keep an eye on, depending on how long your experiments are running.
Okay, makes sense. So they've got the light spectrum dialed in, they found the right seals. Now, what about the distance between the LEDs and the cells themselves? Does that matter?
It definitely does. Remember how the LEDs emit light in a cone shape?
Yeah, I vaguely remember that from physics class, I think.
So their idea was that if they moved the LEDs closer to the bottom of the plate, they could get a more even light distribution across all the wells. And their system was designed to allow them to adjust that distance between 3 millimeters and 20 millimeters.
Okay, so how do they test that out? Just eyeball it?
Oh no, they're way more precise than that. They use this array of 24 photodiode light sensors to measure the light intensity at different distances.
Sounds high-tech. And did it work? Was closer better?
Yep, the data showed that the most even light distribution was at 3 millimeters, which was the closest they could get with their setup. They did find about an 18% difference in light intensity between the inner and outer wells, even at that distance, but they think that might actually be an overestimate due to the limitations of the sensors themselves.
Okay, so closer is better, but there's always a trade-off, right? They mentioned something about the heat again.
Exactly. The closer the LEDs are, the more heat the cultures are exposed to. So for organisms that are super sensitive to temperature, you might need to be a bit more careful. They suggest things like lowering the incubator temperature or increasing the ventilation to compensate.
Always thinking ahead. So light spectrum, seals, distance, all covered. Last thing they tested was the microplates themselves. I was surprised to see they even looked at that.
Yeah, I was too, at first. It seems obvious that you'd want to use clear plates to let the light through, right? But they actually compared clear plates to black plates, which you'd think would block all the light.
So how did that work out? Did the cells in the black plates just wither and die?
Surprisingly, no. They did see a bit of a lag in growth in the black plates at first, but after a few days, the final biomass was pretty similar to the clear plates. And interestingly, the growth in the clear plates was a bit more variable over time, while the black plates had a more consistent growth rate once they got going.
Huh. Okay, so why the difference? Any theories?
Well, they think the lower initial growth in the black plates might be because less light is actually reaching the cells due to lower photopigment absorbance, and the increased variability in the clear plates could be from the light bouncing around inside the plastic due to refraction and internal reflection. It's like a little bisco in there for the algae.
That's kind of cool to think about. So bottom line, is one type of plate better than the other?
It depends. Both clear and black plates can work with this system, but you need to be aware of the potential issues. Black plates might have some edge effects where the outer wells grew a bit slower, and clear plates might have more well-to-well variability.
Got it. So they've designed the system, validated all the components, optimized the configuration, and they showed that it can reliably support photosynthetic growth in microplates. But let's talk about what you can actually do with it, right? The article goes into some pretty cool demonstration use cases.
Yeah, and this is where it really gets exciting, seeing how this technology can be applied to solve real-world research problems in biotech. One of the first things they did was demonstrate how you can use it to manipulate growth conditions in a really high-throughput way. They did this by systematically varying the concentrations of each component in the standard BG11 growth medium for Synechococcus.
BG11, that's like the standard recipe for growing a lot of these cyanobacteria, right?
Yep, it's got 8 different components, and traditionally, if you wanted to figure out which ones are most important for growth, you'd have to make a ton of different media variations by hand and test them one by one. Super time-consuming.
And this is where automation comes in handy, right?
Big time. They used automated liquid handling to create all the different media combinations and then used the lighting system to make sure the growth conditions were identical for all the cultures.
So they were basically able to do in a few hours what might have taken weeks or months to do manually.
Exactly. And they were able to quickly identify which nutrients were most important for growth, like phosphorus, inorganic carbon, and nitrogen.
So that's key for optimizing growth media, figure out which nutrients are limiting, all that good stuff. What else did they show off?
They also demonstrated that the system can be used to test drug sensitivity. They exposed Synechococcus to different antibiotics and were able to accurately reproduce the known sensitivity profiles. So it works for that kind of application too.
Okay, so basic stuff like media optimization and drug screening, very useful. But then they took it a step further and used something called response surface modeling, or RSM, to really fine-tune the media.
Yeah, RSM is a really cool statistical technique that lets you figure out the optimal levels of a bunch of different input variables at the same time to get the best possible outcome. In this case, they wanted to maximize the growth rates of 3 different photosynthetic organisms: Synechococcus elongatus, Chlamydomonas reinhardti, and Nostochaetae.
3 different organisms. Wow, ambitious. And I saw they did this under 2 different CO2 conditions as well. So no CO2 supplementation and then normal atmospheric levels.
Yep, you got it. They used what's called a central composite design, which basically means they tested 3 different levels of each of the 8 BG11 components at both CO2 levels, a ton of different combinations to test. And it really highlights a key point here, which is that high throughput systems like this can dramatically speed up the process of media development.
Which could be a huge bottleneck in biotech, right? Figuring out the perfect recipe to grow your cells can take forever.
Totally, it can be a real pain. So what they found with their RSM analysis was that for both Synechococcus and Chlamydomonas, the concentration of nitrogen, which they added as sodium nitrate, was the most important factor influencing growth. But for Nostoc Haiti, the level of magnesium added as magnesium sulfate was the most critical.
Why the difference for Nostoc?
Well, nostoc is a nitrogen fencer, which means it can pull nitrogen directly out of the air, and that process needs magnesium.
Ah, okay, that makes sense. So once they had all this data, they could basically generate a MADI that predicts the optimal media composition for each organism, right?
Exactly. They built their model, designed some optimized media formulations, and then they actually grew the organisms in flasks using these new recipes to see how well it worked in practice.
And the big question, did the optimized media actually boost growth?
Oh yeah, big time. They saw some pretty significant increases in biomass, anywhere from 38.4% to 61.6% better than the standard BG11 medium. And this was after just 7 days of growth.
Wow, okay, that is a huge improvement. So it's not just about saving time, it's about getting better results, higher yields, all that good stuff. And all that from just optimizing the media.
It's pretty remarkable, right? Imagine what this kind of approach could do for scaling up production of biofuels, bioplastics, pharmaceuticals, all sorts of things.
Definitely. So last but not least, they also use this system to screen for bioactive molecules that can influence algal growth. Sounds like a great way to discover new tools for biotech or maybe even find some compounds with commercial value.
For sure. They screened over 4,200 different compounds to see how they affected the growth of Kinecococcus.
4,200 compounds. That would take forever to do manually.
No kidding. But with this automated setup, they were able to do it in a reasonable amount of time. They did the screen in 3 rounds, first a single replicate for each compound, then they took the hits from that and did triplicate replicates, and finally they narrowed down the best candidates and tested them at a bunch of different concentrations.
So like a funnel getting narrower and narrower until you find the really interesting stuff. What did they find?
They actually found quite a few compounds that had a pretty big impact on growth. One of the most promising was this flavanol called gossypitin, which increased growth by up to 40%.
Gossypitin? I've never heard of that one. Sounds promising, though.
It is. They also found a bunch of compounds that inhibited growth, including gentian violet and disulfiram. Those could be useful for things like controlling harmful algal blooms.
Ah, interesting. Did they do any follow-up on the gossypitin, see how it was actually working?
They did, and this is where it gets kind of weird. When they added gossypitin to the cultures, the color changed dramatically. Synechococcus is normally green, but with gossypitin, it turned almost black.
Black algae, that sounds creepy.
Right. And gossypitin itself is yellow, so it's not just the color of the compound itself. They think what might be happening is that the iron in the growth medium is reacting with the gossypitin and forming some kind of complex that's causing this weird color change. But the really important thing is they were able to validate the growth-enhancing effect of gossypitin in flask cultures, too.
So it wasn't just an artifact of the screening setup?
Exactly. So it's pretty clear that this system is more than just a tool for optimizing known things. It's also a powerful platform for discovering entirely new ways to manipulate and control these organisms.
Really exciting stuff. So to wrap things up, we've gone through the development and validation of this amazing new automated high-throughput lighting system. It's super sensitive, can handle tons of cultures at once, and it plays nicely with all the other robots and equipment you probably already have in your lab.
And most importantly, we've seen how it can be used to optimize growth conditions, identify bioactive compounds, and really accelerate research and development in biotech.
So for all of you out there who are just starting your careers in biotech, think about this. How could a system like this change the way you approach your research? What kind of ambitious experiments could you suddenly tackle that were just too difficult or too expensive to do before?
Exactly. What new possibilities are suddenly within reach?
If you want to dive deeper, definitely check out the original article. It goes into all the technical details, and they've even made all their data and code publicly available on GitHub. There's also a detailed parts list if you're thinking of building your own system.
Or maybe you want to collaborate with them. Who knows?
Lots of possibilities. So that's all the time we have for this deep dive. Thanks for joining us. And until next time, keep pushing the boundaries of biotech.